This is Dr. Mobeen Sayed with one more episode of Long Story Short with DrBeen. Today is the big topic, and that is T regulatory cells or TReg cells. We have done this discussion a tiny bit in our last lecture. We said that T regulatory cells are a cell kind that originates as a result of the start of inflammation or immune system's activity. Mostly naive T cell can be converted into T regulatory cells. These cells are the one that we're going to talk about. T regulatory cells' general function is to inhibit the immune system activity, to reduce the activity and control the immune system back towards homeostasis. And you can realize their importance by realizing that if a baby is born who doesn't have correctly functioning T regulatory cells, then the baby develops, this is a rare disease called IPEX, and the baby would die unless that baby is given stem cells, transplanted stem cells that contain the T regulatory cells. This is how important these cells are. So why does the baby die? When the T regulatory cells are not present or are not functioning correctly, then the body's immune system just goes rampant. And it just keeps trying to produce, or it keeps producing lots of inflammation in the body. It keeps attacking the body. It keeps attacking any pathogens that are coming in, and it does not know how to regulate itself. It does not know how to switch off or go back towards homeostasis. And that inflammatory outcome becomes so intense that it kills the baby. So let's now look at it. This is again our overall bigger picture that we have looked at this before as well. This is just a refresher. This is the innate arm. Innate arm cells, dendritic cells, macrophages, natural killers, neutrophils. Here, let's say this is a dendritic cell. Innate arm cells' function is to, first of all, respond to the antigen. And secondly, their function is to then activate the acquired arm. Now, one part of that activation is production of the T regulatory cells as well, so that when the inflammation has started, when the immune response has started, then we have already created an insurance that will go back to homeostasis because Treg cells are produced. And of course, if this mechanism becomes dysregulated, then we can end up with the chronic infections, or we can end up with the severe infections. So when we talk about chronic inflammations and we talk about dysregulated cells, here is one example of an important cell. So now let's look at how T regulatory cells work. This is a very important topic to look at. First, there are two concepts to keep in mind in context of the T regulatory cells. Number one, T regulatory cells are critical, crucial to create self-tolerance. Self-tolerance means our immune system will tolerate our body tissues and will not attack it. For our immune system, everything is an antigen. Everything is an enemy, including all the tissues of our body. So to stop the immune system from attacking our own body, an important actor are the T regulatory cells. The second important role of T regulatory cells will be that when external triggers, for example, viruses, bacteria, fungi, or allergens, when external immunogens arrive in our body, vaccines are another example, then the inflammation starts or immune response starts. Even controlling that immune response back towards homeostasis is the function of the Tregs as well, or they are at least one major part. So now let's see, how do we make Tregs? So when the baby Treg cells are made, naive T regulatory cells are being made from the stem cells. Stem cells are the parent cells. When the new cell is formed, every T regulatory cell need to have a specific binding region on its T receptors, T cell receptors, TCR. This binding region, imagine it is similar to our arm and hand. So imagine our arm is T cell receptor. We are a T cell, arm is a T cell receptor, and hand is a binding region. Now our hands are very diverse in their functionality, and they can bind or they can grab anything, almost everything. But T regulatory cells can only have one shape, like a wrench, or not even a wrench, a key. They can only have one shape, and they can only bind to anything that can fit into that shape. So now the question is, how do they come up with that shape? These shapes are randomly made for them. That means every T cell has a T cell receptor that has a binding region which is different from the second T cell receptor or T cell. On one T cell, all receptors are the same, but on varying T cells, the binding sites are different. How does that happen? This is something we say recombination, genetic or genomic recombination. So when the T cell is being formed, imagine just like a lottery machine. The genetic components of the T cell, which make the binding region, those components are randomly shuffled and a random number of genetic material is put together to create a pattern. That pattern in turn makes the binding region. That means that as we make billions of these cells and as they all have varying kind of binding regions, some of those binding regions, actually a lot of those binding regions, will be able to bind to our tissue proteins as well, or antigens. Now our immune system has a challenge. We made them, these T cells, we gave them random shapes, but the problem is that these random shapes can allow these T cells to bind to our own tissues and attack them. So we need to now eliminate them. Who? Those T cells that are self-antigen recognizing cells. Self-antigen means the patterns of our own proteins. Those T cells that recognize our tissue, we need to kill them. So what do we do? And before we discuss more, I wanted to also touch upon this concept of immune suppression. For example, let's say steroids do immune suppression versus tolerance, immune tolerance. Tolerance is usually more precise, more specific, and more limited in its behavior. Immune suppression normally is more broad spectrum. So when you are looking at an intervention, when you're looking at a therapy that targets the immune system, ask yourself this question: is this therapy going to be immunosuppressant, like a steroid, or immunotolerance creating, or more specialized, more specific immunomodulator? And we would always like tolerance producing therapies, and you would see in the future we'll have more and more therapies that will be tolerance producing more than suppressing. So now with that little philosophy, here imagine these are two cells in thymus. In the thymus, this cell here, the one that is in gray, this is an antigen presenting cell. Our thymus contains a lot of antigen presenting cells like macrophages and dendritic cells. But these are very special kind of macrophages and dendritic cells. Why are they special? These cells, antigen presenting cells in the thymus, they have a special genetic material open or genetic expression open in them that does not happen in the rest of the body, and that those genes help these cells produce proteins that are called autoimmune regulator proteins, or AIRE. Imagine that these are cells that are sitting in a kitchen and they are making food, and they are able to make a special kind of protein that can be seen in our body, in our tissues. Their job is to create representations, molecular, microscopic, electron microscopic representations of our body proteins. These proteins, when produced, they will help train the T cells. That is one important thing to keep in mind. Second important thing to keep in mind is that whatever antigen means a pattern, whatever antigen is presented in the thymus by these thymus cells, it is considered that these are our self-antigens. That means if a virus goes and hides in thymus, our body, our T cells will think that that virus is our self. So anything that is present in the thymus is considered self. Why? So one, we just saw that these cells can actually produce patterns of proteins that are similar to the proteins found in the cell, in the body. Secondly, most of the time, the infections that occur, the viruses, the fungi, the bacteria, they usually don't get a chance to end up in thymus. For example, let's say I have an injury on my finger, and from that injury, there are some bacteria that have entered this area. That is a local infection. Local macrophages, dendritic cells, immune system cells will attack that pathogen, pick it up, go to the lymph node, and then start working with it. But hopefully, I will not develop bad enough infection. And if the normal situation is present, then that infection will be taken care of, and nothing from that bacteria or virus or fungus will get into my thymus. So because of these reasons, these antigen-presenting cells know that when we are presenting an antigen to a naive baby T cell, that antigen can be assumed to be our self-antigen. Then, if this baby naive T cell reacts to this self-antigen, that means this is a bad T cell. It is going to go out in the tissue and attack our tissues because it is reacting to a pattern of our tissue. So this is the training that is happening in the thymus. And now this cell, if it reacts to our own antigen, then this cell has two choices now. But before that, if this cell not reacts at all, it does not even engage in this activity, then that cell is killed because it's not going to engage in any activity in the cells in the body at all. So first, this cell has to say that, hey, I will engage. Once it is engaged, then we see, is it going to react to the body's own patterns? If it is going to do that, then in the thymus, this naive T cell is given two choices. What are the two choices? One is this T cell has to die, called negative selection. Apoptosis has to be triggered. That means a message is given to this T cell, naive T cell, this baby cell, just got made a couple of days ago. It's in thymus, and it got this message that, hey, you are not a good cell, kill yourself. That is apoptosis. And this cell would trigger internal caspases and ubiquitin proteins, and the result is that cell would die. There is another choice given to this cell. That choice is given, and you can ask me that, how is the cell given a choice? So is this really this dialogue going on? And I'll tell you how. There is another choice given to this cell, and that is, hey cell, you know how to accidentally, we're not happy about it, that you know how to bind with our own self-antigens. You recognize as ourself as an antigen. So it's not a good thing, but you do that. And we need cells who can do that so that you go out in the field, that is the body of the person. And if you can recognize self and you see any other cell attacking the self, then tell that cell, the other cell, to say, stop it. This is self. I know it is our own tissue. Stop destroying it. So become a regulator cell. Now, what does this mean? How can this cell become regulator cell? I mean, no cell is going to say, all right, kill me, but there are going to be some cells that would die. Here is how. There is a lot of interleukin-2 produced in thymus. If a cell, T cell, naive T cell, T helper cell, reacts to our self and also responds to a lot of interleukin-2, then that cell can become T regulator cell. On the other hand, if a helper naive T cell reacts to our self antigen but does not react to interleukin-2, then that cell has to go through apoptosis and die. So interleukin-2 receptor density and response to interleukin-2 is an important player or factor in one of these two fates. So now we made a T cell, helper T cell. The regulator cells are actually a type of helper cell, as I said before. We made a cell that went into thymus, got trained, got found out that it is self-reacting. We said, hey, do you want to be a regulator? If so, react to interleukin-2. It said yes, and now it is a regulator T cell. So I made this cartoon a few days ago that a naive helper T cell goes to therapist. And so here is the naive T cell sitting with the therapy cell and saying, I had a nightmare that I am in thymus for training and I am self-reactive because these poor little cells can be killed. Okay, so now, how do T regulatory cells actually help with the regulation? This is a huge, big science, and I'm going to try to encapsulate the summary of that in this one diagram. So it looks busy, but you would actually see if we go methodically over it, it will almost put all of the important functions of regulator cells in this one area. So here, let's start from this part, number one. This is a T regulator cell. It is now out in the body. it has a special protein on it called CTLA4, cytotoxic lymphocyte antigen A4 or antigen 4. Why is it called CTLA4? This protein was very first time observed on a cytotoxic cell. So it was named as CTLA4, although then it was found out that CTLA4 is present on, for example, regulator cells as well, which are helper cells. So CTLA4 proteins are present on regulator cells. Now keep that in mind, and I'm going to tell you a story which is very important and very interesting as well. It's a love story. Hopefully you'll see there are some love stories that would go in a bad ending. This seems to be okay. So here in our tissue, imagine this, let's say this is our finger. In our tissue, an antigen-presenting cell, innate arm cell, a macrophage or a dendritic cell, incorrectly tried to present our self-antigen to immune system. Now one more clarity over here. These cells don't have brains to say don't present self and only work with viruses or bacteria or fungi or allergens. The function of these cells is they're going to pick up whatever is in front of them and try to present that as an antigen. It is the remaining cell's responsibility to not react to that antigen if that antigen is our self-antigen. And that is where regulator cell is going to come in. So that's why I have made the regulator cell here. Okay, so back here, this antigen presenting cell is sitting in my finger and it picked up a microscopic piece of my muscle, for example, or connective tissue, and tried to present that to another T cell that is also here in the vicinity. Let's say this is also a T helper cell. So here you are seeing an interaction between them. This is the MHC2 molecule, major histocompatibility class 2 molecule. On that is this little piece of my protein. And this is a T cell, helper naive T cell, that has this T cell receptor. And they are connecting here. This is called antigen presentation. Once this antigen presentation is done, ideally this naive T cell will become active and inflammatory response would start. But you know it will be really bad if the inflammatory response starts like this. The whole body will be under attack. That will be like IPAX and we would die immediately. So this cell, naive T cell, says, I'm not going to become active like this. Give me a co-stimulation. Do a double check with me. And the co-stimulation is done here where there is another protein on the naive T cell called CD28. And that binds with another protein on the antigen presenting cell called B7. B7 and CD28 also have to bind for double checking that, hey, go ahead with the destruction. Even then it is not sufficient. There has to be chemical substances released from antigen presenting cells that would have to give hint to the naive T cell to say, I am confirming this is a bad thing, we need to attack. For example, interleukin 12, interleukin 1, interleukin 2, interleukin 8, and so on. The point is, there is double checking and triple checking. In the medical sciences, in immunology, we call it co-stimulation. That this is the stimulation, but there needs to be co-stimulation. With the hugs, there need to be kisses. And only then these cells will become active. Now, this T cell, the regulator T cell, is observing these guys. And remember, he made a promise to us that if he saw some cells trying to attack self, then he will intervene, the cell, it will intervene and stop these cells from destroying the tissue or reacting to the tissue. So how does that happen? So this T regulator cell is present in this area as well. It saw that this is happening. How did it see that this is happening? Because the same antigen presentation that is happening from this antigen presenting cell to this CD4, that cell is also presenting that antigen to this guy as well. So this presentation tells this regulator cell that this antigen presenting cell is presenting self. So guess what this regulator cell starts doing? It's so funny. What it does is it extrudes its CTLA4 molecule, and that molecule is called an endocytic molecule. A molecule with which if something is bound, then the cell will eat it. Imagine my hand. Whatever you put on my hand, I'll eat it. Or I'll drink it. Whatever glass of fluid you give me, I'll drink it. Then my hand will be called an endocytic molecule. Whatever you place there, it's gone, eaten up. So this regulator cell, these are such beautiful mechanisms, I get amazed. This regulator cell attaches a CTLA4 to the B7 on the antigen presenting cell and nibbles away, plucks away the B7 and eats it up. So soon it removes the B7s from antigen presenting cells. It nibbles them, it grazes them away. Of course, the actual term is it endocytoses them. When one cell of our body nibbles away proteins from another cell, we call that transendocytosis. So this CTLA4 is an endocytic molecule. This process is transendocytosis. The result is that on the antigen presenting cell, B cell molecules reduce in number. Now, why is that interesting? Here is why. Remember, if you go back to this one, for the co-stimulation, for the immune system to start reacting to this antigen, which is actually my finger's connective tissue, there has to be co-stimulation of B7 and CD28. But if the B7 on the antigen presenting cell is gone, it has been nibbled away by the regulator cells, then who is going to connect here? No one. And the antigen, the CD4 cell, will develop energy, ANERGY. It would develop tolerance. It would say to this antigen presenting cell that, hey, thank you very much for presenting me this antigen. I think you are not able to co-stimulate me with B7. Must be another regulator cell in the vicinity that has grazed away, endocytosed your B7s. I should not become active. This is how regulator cell protects us. This is an example of regulator cell producing self-tolerance. So that's one mechanism. Second mechanism, if you arrive on the left side, regulator T cells can produce interleukin 10. Remember, T helper 2 can produce interleukin 10s. These dendritic cells or antigen presenting cells themselves can produce interleukin 10. And of course, regulator T cells produce interleukin 10, chemical substances. What is the function of the interleukin 10? The function of interleukin 10 is to act on the macrophages and dendritic cells and reduce their function. Remember, we are talking about regulator T cells. They're going to regulate things. So what is a function reduction? That is, number one, on the macrophages and dendritic cells, which are innate arms antigen presenting cells, interleukin 12 production is reduced. Interleukin 12 production is very important for activity of the immune system to continue and to expand. So when interleukin 12 production is reduced, the innate arm and acquired arm would start calming down, plus the macrophages and dendritic cells would become less active. So who is doing that? T regulator cell. How? By producing interleukin 10 that acts on macrophages and dendritic cells and reduces the production of interleukin 12, which in turn then calms them down and calms a lot of other immune system actions. Then major histocompatibility complex number 2 production is reduced. What does that mean? If I zoom in once again, remember I said that this antigen presenting cell, which has plucked away my antigen, my connective tissue part, and trying to pass it on to the CD4 to say, attack this thing. This little arm of the antigen presenting cell is made up of a protein which is called MHC2, major histocompatibility complex number 2. So of course, if this MHC2 molecule is less produced, then this cell would have less arms to present the antigens. And that would mean less presentation of the antigen, less reaction to the antigen, less immune responses. So that is another way to downregulate the function of the innate arm. And you're seeing that this is also downregulating the function of acquired arm, because acquired arm co-stimulation, acquired arm antigen presentation is going down. Okay, what else happens? I think you would love this part. Production of interleukin 2 activates the immune system. However, T regulator cell, they consume the interleukin 2. They remove the interleukin 2 from the environment. So imagine that you are an immune cell, dendritic cell, helper cell, cytotoxic cell. You are standing somewhere where there is water, fluid on the floor, and that water is IL-2. So higher the level of the water, the more active you are. And if there comes a machine that sucks away that water, then you are less active. This is what these regulator cells do. They take the interleukin 2 from the environment and they consume it. Remember, one of their property in the thymus was that they consume a lot of interleukin 2. They have a lot of interleukin 2 receptors on them. They have so much interleukin 2 receptors on them that these cells are recognized by the density of the interleukin 2 receptors, or CD25 we call it. This is why these cells are called CD25 positive. Beautiful science, immunology, or our body. So, because of those extra receptors for interleukin 2, these cells are going to pick up interleukin 2 from the environment, consume it, remove it, which means that for the remaining cells, the amount of interleukin 2 is less. That means the immune system cannot stay as active as possible. Interleukin 2 is the fuel for their activity. So this is one more way of regulating. Then T regulator cells produce tumor growth factor beta. Now, tumor growth factor beta is produced by many cells, including T regulator cells. Once TGF beta is present, what it does is it inhibits the TGF beta, inhibits other T cells. For example, if you put TGF beta on this T cell will not work as much. If you put TGF beta on the macrophages and dendritic cells, they would not work as much. Plus, TGF beta causes class switching of the B cells towards IgA. There are many, many functions, and there are controlling functions. So Treg also produces TGF beta, which also helps bring homeostasis in the immune system. So with these main functions, which I have encapsulated in one area, let's go to the few more pointers about regulator T cells. Regulator T cells were suspected to be present in our T cells from 70s. Scientists suspected that there are some cells in the T cells which are able to control the other cells and bring the function of the other cells down. But they did not know which these T cells are. In 1990 and onwards, scientists for the first time were able to purify, that is separate them, purify them, analyze them, study them, and classify them, categorize them. So in the sciences, this is kind of relatively interesting new science. These cells have markers on them, just like all helper cells are called CD4. Helper T cells are called CD4 positive. Cytotoxic T cells are called CD8 positive. These cells, T regulator cells, are actually helper cells, so they are CD4 positive. But how do you differentiate them from naive T helper cell, other naive T helper cells, or T helper 1 cell or T helper 2 cell? Here is how you differentiate them. Number one, these regulator T cells have a lots of interleukin 2 receptors on them. Interleukin 2 receptors are designated as CD25 proteins, cluster of designation number 25. So these regulator T cells are called CD25 positive because they have a lot to do with interleukin 2. Interleukin 2 activates these cells. They use interleukin 2 to function. Remember, if the interleukin 2 is not present or less present, then T regulator function is going to go down. That is why, for proper immune behavior, interleukin 2's correct amounts is paramount. Less interleukin 2 will mean less T regulatory function, will mean enhanced inflammations, chronic inflammations, sustained inflammations, intense inflammation. More interleukin 2 will mean more regulator activity. That means suppressed immune system, which may give rise or a chance to the infections and other things or cancerous cells to develop more. So interleukin 2's balance is very important. So these cells now, CD4 positive, that is helper cell, plus CD25 positive, interleukin 2 receptor. Then these cells inside of their cytoplasm, they have FOXP3 protein as well. So these cells are also called CD4 positive, CD25 positive, and FOXP3 positive. Although there are many phenotypes, there are many variations of these cells. But this is generally the bigger picture. Now FOXP3, this protein that is inside, that has an interesting function as well. Most of the activation signals to a regulator cell, T regulator cells, eventually go and activate FOXP3. And they finally work through FOXP3. FOXP3 is the final common pathway here. So when the FOXP3 is activated, it is a transcription factor. Transcription factor means a protein which when activated can go to the nucleus or can influence other proteins that would eventually cause a gene expression and transcription of messenger RNA and then production of proteins or responses. FOXP3 is essential, so please hold on to this thought. If you forget everything, please remember interleukin 2 and FOXP3 are essential for the generation of a T cell, for the survival of a T cell. If a T regulator cell, I shouldn't say just any T cell, regulator cells, if a T regulator cell doesn't have sufficient FOXP3 or doesn't have IL-2 activating the FOXP3, that T cell would die. So continuing it to survive, we have to provide IL-2 and FOXP3 activity. So FOXP3 is necessary for generation of the T regulator cell, for survival of the T regulator cell, and for the function and growth of the T regulator cell. What does growth mean? Making more copies of these cells. So if IL-2 is not present, if FOXP3 is not present or not functioning correctly, we are in trouble. So development, function is very important. And as I said before as well, if there is a mutation in rare cases, in some children, there is a mutation of FOXP3 producing gene. So FOXP3 is either not produced or produced in a defective way. So T regulator cells do not work very correctly. And this child is going to develop overwhelming infections and inflammations and autoimmune disorders and die. So this child has to be given stem cells that would produce correct T regulator cells. And now the last part of the talk today, some phenotypical markers, we just talked about them. These cells are CD4 positive. These are FOXP3 positive. These are CD25 high. That means compared to other cells, the CD25, which is IL-2 receptor, the density of that is high on these cells. They also have an increased number of CTLA4, as I discussed above. They use less interleukin 7, they use more interleukin 2 compared to other cells. Now, interleukin 2, as I just discussed, works through FOXP3. So that means interleukin 2 is going to be important for generation, survival, regulation, function of the regulator cell. Interleukin 2 works through JAK-STAT pathway. So it influences the STATs, which then influence the FOXP3, which in turn has or promotes the activity of the regulator cells. The regulator cell, these are CD4 helper cells, right? So they come in naive state when they are freshly born. They are in active state when they are actually taking part in an inflammatory behavior. And then they become memory as well, just like other cells become memory. So this is a very important thing. A bad memory T regulator cell would propagate bad control, which will mean possibility of chronic inflammation, chronic hypoinflammation or hyperinflammation. Then these regulator cells are also based on their location called thymic regulators or peripheral. Either they're present in the thymus or in the tissues. They are also called, based on their location, lymphoid or non-lymphoid. Lymphoid means those T regulator cells that are present in the lymph node or other lymphoid tissue clusters, and non-lymphoid mean the rest of the body. And finally, T regulator cells are of course produced in general. They are being produced right now in me and you. But when there is an inflammation that happens, when there is an infection that happens, during the inflammatory processes, new T regulator cells are formed as well, whose job is to provide homeostasis to this new inflammation. What does that mean? With every war, controllers are made during the war to say, your job, controller, is to control this war. And as we said before as well, some T regulator cells need TGF-beta, which the TGF-beta also helps stimulate FOXP3. And if the TGF-beta is less, then the FOXP3 function is less, and then the generation, survival, growth, and function of T regulator cell goes down. That creates immune dysregulation, which then creates huge issues. Less inflammation when we need more, or more inflammation when we need less, or body's inability to go back to the homeostatic state of inflammation, meaning the no inflammation state, and body just gets stuck in a bad inflammatory state. So this is about the T regulator cells. Thank you very much for listening in. I would see you with the next episode. Bye-bye for now.